carbon dioxide release is from glucose, and this was not affected, and how effectively glutamate production is from alpha-ketoglutarate. And alpha-ketoglutarate and glutamate are in equilibrium, so this is a good marker, a marker that can actually characterize the TCA cycle function in tumor cells. And as I showed you, the only responsive cell line as far as TCA cycle flux goes was this MCF7 breast cancer cell line. So obviously we were more interested than finding out what is the common metabolic change that we can observe in all tumor cell lines. And interestingly, there was a unanimous yet different species of fatty acid synthesis were decreased, but in, for example, in mesopancreatic cancer cell lines, there was a decrease in palmitate as well as in lignoserate synthesis, and this is a saturated long-chain fatty acid participating in membrane synthesis. And there was also a decrease in, interestingly, not in fatty acid, but in cholesterol de novo synthesis in the MCF7 breast tumor cell lines. And just to show you how effective the treatment was, in mesopancreatic adenocarcinoma cell lines, the lignoserate rate of labeling, which you can see here by numbers, in the 72 hours experiment, about 60% of lignoserate was labeled in mesopancreatic cancer cell lines. It decreased dose dependently after 50 and 25 ppm DDW water. In mesopancreatic cell lines also palmitate synthesis was down after 25 ppm water significantly, and these long-chain saturated fatty acids are membrane components, and in the MCF7 breast cell line, there was a decrease in cholesterol synthesis. Again, this was a dose, at least the higher depletion induced a more robust effect in decreasing cholesterol synthesis. So truly, the question of how would decreased long-chain saturated fatty acid affect tumor cell metabolism or rate of proliferation is probably a more complex question, and obviously this study is sufficient to think more about how to go after these mechanisms, but definitely all these fatty acid species that I've been talking about are very important and very critical fatty acids for cell proliferation, just simply because they are the source of cell membranes. And interestingly, some of the long-chain saturated ones and cholesterol in membrane, their function is to actually anchor certain proteins and make the membranes more rigid as far as allowing the cell membrane proteins to function properly, while other parts of the membrane more prone to contain palmitate or oleate, which are actually with less chain length and more oily, meaning that actually the membrane is more fluid. And there might be important roles of these fatty acids in nuclear membrane synthesis, which is the first step of DNA replication and packaging before the cell proliferation process actually gets completed. So, and just looking at, again, for fatty acid synthesis, there is a citrate shuttle. Citrate shuttle will actually generate oxaloacetate, but the label part is carried towards cholesterol synthesis, and this is how cholesterol gets labeled. This was decreased in MCF7 cells, and as far as long-chain saturated fatty acids, they may become all labeled, meaning that if you use a fairly high enrichment of glucose tracer, then your fatty acids can be labeled, and then completely almost, or if you use a tracer which is a uniformly labeled fatty acid, then you can obviously follow up all the scenarios that we have observed with glucose. So there are some new ideas and studies that are obviously capable of focusing on just fatty acid production synthesis, which seems to be the common effect in these tumor cell lines as far as the metabolic effect of deuterium depletion goes. So as a short conclusion, we found that, in fact, decreased deuterium to hydrogen ratios seem to regulate sterol synthesis as well as fatty acid precursor synthesis. And this may affect the rate of divisions of cellular systems, or explain why and how, from the mechanism point of view, maybe deuterium depleted water is able to affect tumor cell physiology in the desirable manner. Again, obviously these studies need to be followed up with certain tracers and specifically finding out what the mechanisms are and what are the good markers that will differentiate tumor cell phenotype from another tumor cell. Truly, what is the optimum ratio of DD depletion, when we need to switch or increase depletion to actually overcome the obvious adaptive effects of the cells or metabolic networks to deuterium depletion. But in fact, we believe that this is a good start to find out exactly how to advance in the field and how to use DDW as an effective treatment in cancer. Thank you very much. So that said, any questions from the audience? Go
ahead. Thank you for your presentation. If you said that finally the DDW can consider as the inhibitory factor of the fatty acid synthesis. Can we say that somehow we could reduce the fatty acid synthesis?
I think fatty acid synthase is a huge complex. It's a protein that swims in the cytoplasm. It's actually anchored to the smooth endoplasmic reticulum. And what happened, or what may happen, is to explain these effects, we need to bring in metabolic control analysis, which will tell you exactly how the enzymes kinetics, including elasticity, changes when deuterium is replaced with light hydrogens, meaning that probably what happens is that this enzyme holds hydrogen in one hand and has the acetate units which need to be actually brought into in a certain kinetics to this process proceed appropriately. So this may not be an enzyme inhibitor, but enzyme function inhibitor, meaning that it specifically inhibits this process. Enzyme activity may not change, or even protein expression of the enzyme may not change, yet the function is the enzyme is definitely affected.
Thank you. So the question would be, if we take an anti-cancer drug, and if you should compare the results that you can get with the fluxes and all these things, to compare that effect what an anti-cancer drug can cause with the effect of the deuterium depleted water, what would you say that what is the ratio? How strong effect can you consider what we can say with DDW?
Yeah, it's a very good question. Actually, C75, which is a Merck drug, it is developed against fatty acid synthase. So it is, in fact, a targeted pharmaceutical compound for inhibiting fatty acid synthase, because actually fatty acid synthase is now a very prominent target in cancer research. If you look at C75 and compare the effect with DDW, the 25 ppm is just as effective as C75 at 100 micromolar range. Now, interestingly, if you use natural products of fatty acid inhibitors, just like there's luteolin, which is a green tea product, surprisingly, using the same dose head-to-head comparison with C75 luteolin or DDW, they have practically the same effect as far as inhibiting. So their efficacy is very comparable with pharmaceutical drugs. Again, the mechanism is very different as far as how they achieve this phenomenon. Obviously, we need to know exactly how the kinetics, enzyme kinetics, adapt to these different treatment scenarios. But in the first 72 hours run in cell cultures in vitro, we can show the same efficacy of DDW as compared with pharmaceutical drugs that are targeted against fatty acid synthase. Any other question?